Hello, my name is David Bressler, and I'm presenting Drosophila melanogaster with varying polyglutamine repeats identified to impair locomotive function. So first, let me give a little background on what we were researching and why it's important. So what we're researching is Huntington's disease, and this is a rare neurodegenerative disease that currently has no cure and is fatal. So the neurodegeneration of this disease is important because we need to understand how motor impairment occurs and the system behind it. So Huntington's has been found to be caused by excessive glutamine or CAG repeats on the first exon of the HTT gene, and this gene codes for the Huntington protein. So these CAG repeats, what they do is if they're in excess, they cause this protein to aggregate, which doesn't allow it to perform its function, causing the degradation of the neuronal cells. So what we're interested in is studying if more increased CAG repeats increases the amount of motor impairment. So what we did is we used the Drosophila melanogaster as a fly model, and this is because we know the chromosomes that it has, and the fly is also a good model because it has a short grow time, and we understand how the systems work within it. We also use the ELAV-GAL4 UAS driver system, and what this did is the ELAV-GAL4 driver allowed only the neuronal cells to be targeted, and the ELAV driver made the GAL4 gene create GAL4 protein, and the GAL4 protein then bound to the UAS system, which promoted the expression of however many CAG repeats we had placed into these flies. So we had two separate flies, one set with 16 repeats and another set with 128 repeats. The 16 repeats were UAS Q16 and the 128 repeats were UAS Q128. So we did make crosses. So we made a cross with ELAV GAL4 males with UAS Q16 females. So we used one set of these, which was ELAV GAL4 UAS Q16 females. Then another set was ELAV GAL4 crossed with UAS Q128 females. And so from here we actually used two groups. We used the ELAV GAL4 UAS Q128 females as the other test group, and the UAS Q128 males as our control, as these would not have any CAG repeats in them. So what we did is we performed a negative geotaxis assay, and essentially what this was, we put one of these sets into a graduated cylinder, we knocked them down, and we gave them 20 seconds to climb to the top. Then we took a photo. And in the diagram is an example of what that could have looked like after 20 seconds. So what we considered a successful test is all the flies that reached above 40 milliliters were considered to have passed the geotaxis assay. So as our results show, the controls had performed the best with 52% reaching the top, and this table we ran 3 trials and we averaged the data and got percentages. So 52% of the males reached the top with 8% standard deviation, while the Q128 performed the worst with only 23% reaching the top. So this actually suggests that the more CAG repeats that an organism has, the more locomotive impairment that it will get, because the UAS Q128 females had the most repeats and performed the worst, while the control males had no repeats and did perform the best. So since Huntington's disease is a age progressive disorder, we would also like to, in further research, test each stage of its development, so every few days, to see how the progression of the disease is actually happening. So we would like to thank Dan Zhao, Gabby Calcano, Kim Piasta, and Lindsay Mermanish for making this experiment possible. Thank you.